million in tissue.
Next main challenges of gene therapy is immune response. Our immune system are very good at fighting of intruders such as bacteria and viruses. Gene delivery vectors must be able to avoid body natural surveillance system. An unwelcome immune response could cause serious illnesses or even death. For instance, the story of Jesse Gelsinger, who had a rare liver disorder that participated in 1999,
disrupting important genes in target cells. A good gene therapy is one that will last. Between 1999 and 2006, researchers tested a gene therapy treatment that would restore the function of crucial gene, gamma C, and cells of the immune system. The treatment appeared very successful, restoring immune function to most of the children who received it.
One of the challenges of gene therapy is the commercial viability. Many genetics disorders that can be potentially be treated with gene therapy are extremely rare, some affecting just one person out of a 1 million. Just take note of that. Gene therapy could be life-saving for these patients, but the high cost of developing a treatment makes it an appealing prospect for pharmaceutical companies.
Now let's talk about risks to society and direct risks to research participants undergoing stem cell transplantation. Although there may be direct risks to research participants in stem cell transplantation research, there are also social risks. And one major social risk in using embryonic stem cells as a source of neurotransplantation is that new embryonic stem cell lines would have to be created. The derivation of human embryonic stem cells results in the destruction of the embryo, presenting an ethical dilemma for those that ascribe a considerable degree of personhood to embryos.
Other risk of stem cells research is immune rejection of transplanted stem cells. Cyclosporine is a widely prescribed immunosuppressive drug that prevents rejection after transplantation of solid organs or bone marrow, with long-term use shown to cause hepatotoxicity, nephrotoxicity, hypertension, and immune suppression.
The rest of gene therapy is neurosurgical hemorrhage and post-operative infection. It is estimated that stereotactic neurosurgery used in the transplantation of FVM tissue may be associated with a 3% risk of intracerebral hemorrhage and post-operative infection,
Inappropriate stem cell migration, and neurological complications. Research participants receiving stem cell transplants may be at risk of experiencing neurological complications owing to stem cell migration from the graft site to inappropriate regions of the brain.
One challenge facing researchers is how to control and regulate the process to successfully trigger differentiation into the desired cell type after the stem cells have been isolated.
Stem cell treatments include new technologies and therapies that aim to replace damaged tissues and cells in order to treat disease or injury by Landval and Kukaya 2006.
The first challenge researchers face when considering stem cell treatment is to understand the mechanisms by which stem cells function in the injured microenvironment using animal models, and then to translate the results of these studies to humans.
The goal of stem cell treatments is to publish all findings and experiences of stem cell treatments, experimentally and clinically, and to open up new possibilities for the treatment of various diseases such as Alzheimer's diseases, traumatic brain injury, heart diseases, diabetes, cancer, and etc., that we may promote health and extend the human lifespan.